Hello dear students, welcome back to online CET coaching classes on the biology, and today in this video I am continuing the next part of first PU chapter in the human physiology, that is digestion and absorption. In the previous video I explained about the digestion of carbohydrates, digestion of proteins, and what are the different enzymes required for the digestion of carbohydrates and proteins. The carbohydrates are digested by carbohydrates and proteins are digested by proteases. The digestion of carbohydrates is confined or it occurs in the mouth and it is completed in the small intestine, whereas digestion of the proteins, it begins in the stomach and it is completed in the small intestine. And today in this video I am going to explain about the digestion of fats, digestion of nucleic acids, and where exactly the absorption occurs. What are the different methods of absorption of the digested food material, and in different parts of the alimentary canal the different substances are absorbed along with the caloric value of the absorption and digestion of different energy molecules. So let us continue with the digestion of fats. Fat digestion, it starts in the stomach. Stomach, it stores the food for longer period of time and the digestion of the fat is completed in the small intestine. Almost all the food particles, not almost all the food particles, whether it is carbohydrate, it is protein, it is fat or it is nucleic acid, all these molecules are finally completely digested in the small intestine. And point to remember is that the enzymes involved in the fat digestion are known as lipases, carbohydrates, proteases, lipases and nucleases. So lipases are the enzymes which are involved in the digestion of lipids or the fat molecules. So what happens in the stomach to the fat molecule and how it is completely digested in the small intestine. In the stomach, the gastric juice secreted from gastric glands of the stomach, they contain a small amount of gastric lipase. So the first enzyme with respect to the digestion of fats is gastric lipase, and this converts some fats into monoglycerides and fatty acid. Partial breakdown, there will be no complete digestion because for the complete digestion of the fats, emulsification is basic required process. After emulsification only the fat molecules are completely digested into the fatty acid and glycerol. But gastric lipase, it is acting on the fat molecule before emulsification, because emulsification is carried out by the bile salts and bile salts are present in the bile juice, and bile juice, it acts on the partially digested food in the duodenum part of small intestine, so that there is no complete conversion or the breakdown of fats, even the enzyme is present in the stomach. The gastric juice of the stomach contains gastric lipase and this converts some fat into monoglyceride and fatty acid. Then have a look what happens in the small intestine. In the small intestine, the bile juice acts on the fat molecule. So bile juice has bile salts. I hope you remember the names of bile salts. sodium glycolate and taurocolate. So glycolate and taurocolate of sodium and potassium are the bile salts which are present in the bile juice. These bile salts break down large fat globules into smaller globules so that pancreatic enzymes can easily act on them. This process is known as emulsification of the fats, and bile juice also makes the medium alkaline and activate the pancreatic lipase. This is the very important part with respect to the digestion of fats. For the complete digestion of the fats, the required process is emulsification. What is this emulsification? The bile salts sodium glycolate and taurocolate, potassium glycolate and taurocolate. So these are the bile salts along with the bile pigments, bilirubin and bilirubinin, but here I am talking about bile salts. They are glycolate and taurocolate of sodium and potassium. These bile salts, they combine with the larger fat globules, and these larger fat globules are converted or broken into smaller fat globules. The process is called emulsification. After emulsification, the fat is now referred as the emulsified fat, and emulsification provides larger area for the breakdown of fat molecules, and this is further carried out by pancreatic lipase. Here you try to remember what is emulsification of fat. The larger fat globules, they are broken down by the bile salts into smaller fat globules. The process is called emulsification. Remember, emulsification is a compulsorily required process for the digestion of fats. Then how exactly it occurs? Here is the image to show the emulsification. Yes, this is larger fat globule. In the image, this structure represents larger fat globule, and this is combining with the bile salts. So two portions are there in the bile salts that I would like to highlight: hydrophobic and hydrophilic. Hydrophilic part is always outwardly directed when it combines with the fat globule, because hydrophilic part, it absorbs the water or it attracts the water, and hydrophobic part, it repels the water. And remember, digestion or breakdown of the molecules by the enzymes is hydrolytic process. That means it requires the involvement of water, so that hydrophilic part of the bile salt, it is outwardly directed when it is attaching or combining with the smaller fat globules. This larger fat globule, by mixing with the bile salt, it is converted or it is broken down into smaller fat globules, and these smaller fat globules are referred as emulsified fat droplets or emulsified fat or micelles. And further, these emulsified fat droplets are completely digested by pancreatic lipase and gets converted into the fatty acid and glycerol. That is why the emulsification of the fat globule is much more essential process in the digestion of fat. Hope this is clear for you. So next, very important point, bile helps in the digestion of fat by emulsification. Bile helps in the digestion of fat by emulsification. See, bile juice do not contain any enzyme. any type of the enzyme is absent in the bile juice, and bile juice is the one digestive juice which is not having any digestive enzymes, but the salts of the bile juice are essential for the emulsification of the fat, which is further resulting in complete digestion of the fat molecule. So conversion of fat into micelles, that means smaller particles or tiny droplets, is nothing but emulsification, and that emulsification is carried out with the help of bile salts. Then question is, what is the significance of emulsification? Before this one, before studying of the digestion of the fat, you have studied about digestion of carbohydrates and proteins, but in that digestion process, like this, there will be no processes like this emulsification or something else. Only inactive form of the enzymes are converted into active forms with the help of HCl as well as enterokinase and trypsin. But why the emulsification is necessary or essential for the digestion of fat? Here is the answer. Emulsification provides large surface area for the action of lipase on the fat. So all these are the open parts where the enzyme can act on the small emulsified fat globule, and further it will be finally converted into fatty acid and glycerol. The emulsification provides large surface area for the action of lipase, and lipase is nothing but enzyme acting on the lipids. For this reason only the fat molecule is emulsified with the help of bile salts. Then bile also activates lipase. So bile juice keeps the environment alkaline, and alkaline conditions always activate the lipase enzyme, which is further acting on the lipid molecules. This is how emulsification is carried out by the bile salts, and emulsification provides large surface area for the action of the lipase, which helps in complete breakdown of the fat molecules into fatty acids and glycerol. So moving to the next point, what happens? Action of pancreatic enzyme. Pancreatic juice also contains the lipid digesting enzyme called pancreatic lipase, and pancreatic lipase is present in the pancreatic juice. Along with intestinal lipase present in the intestinal juice, both of them hydrolyze the emulsified fat molecules into triglycerides, diglycerides and monoglycerides, and ultimately they are converted into glycerol. See, in the carbohydrates like polysaccharides, disaccharides and monosaccharides, likewise in the lipid molecule triglycerides, diglycerides and monoglycerides. Final action is carried out by two important enzymes. One is pancreatic lipase and another is intestinal lipase. So only three enzymes are necessary for the complete breakdown of lipids. One is gastric lipase, which is present in the gastric juice. Another is pancreatic lipase, which is present in the pancreatic juice, and one more is intestinal lipase, which is present in the intestinal juice. Three enzymes along with the bile salts, they help in complete digestion of the lipid molecules in the alimentary canal. So here is the final representation. What happens to the fat molecule consumed along with the regular diet? So fats in the presence of pancreatic lipase. So pancreatic lipase is acting, means already fat molecules are combined with the bile salts and it is converted into triglyceride and diglycerides. Diglycerides and monoglycerides, again in the presence of lipase, they are finally broken down into fatty acid and glycerol. The pancreatic lipase is the principal enzyme for the digestion of fat. Remember, after emulsification of the fat, the complete breakdown of the lipid molecule into fatty acid and glycerol, it is carried out by the pancreatic lipase. So pancreatic lipase is the principal enzyme, that means main enzyme which is involved in the digestion of fat molecules. So moving to the representation, fats in the presence of lipase, they are converted into the smaller molecules called diglycerides in the presence of lipase, and diglycerides are also in the presence of lipase, they are converted into monoglycerides. Both diglycerides and monoglycerides, again with the action of lipases. Among these, the simplest one are the monoglycerides. Triglycerides are complex one. Diglycerides are also complex than that of monoglycerides. The most simple form are the monoglycerides. So both diglycerides and monoglycerides are finally broken down into the fatty acid and glycerol with the action of lipase. Then what is the function of intestinal lipase? The intestinal lipase, it converts remaining fats into monoglycerides and fatty acid. If any fat molecule is left unconverted, such fat molecules are converted into monoglycerides and fatty acids by the action of intestinal lipase. So this is about the digestion of fat, which is started in the stomach and completed in the small intestine. The enzyme of the stomach acting on the fat molecule or the lipid is gastric lipase. In the duodenum, the bile salts combine with the fat globules. Larger fat globules are converted into smaller fat globules. The process is called emulsification, and smaller fat globules are called emulsified fat, or they are also known as micelles. Emulsification provides a large surface area for the action of lipase, and the principal enzyme involved in the complete digestion of the lipase is pancreatic lipase, and the end products of fat digestion are fatty acid and glycerol. So moving to next one, it is about digestion of nucleic acids. What are these nucleic acids? These are the biomolecules. They function as a genetic material and also controls the protein synthesis, and they are nothing but DNA and RNA. You may ask the question whether we eat the DNA or RNA. Is it there in our food? Yes, all the plant cells, all the animal cells, whether the individual is vegetarian or the individual is non-vegetarian, irrespective of that, whatever the food we consume as our regular diet, they are either from the plant origin, maximum are the plant origin sources, and some are the animal sources. All the content includes nucleic acids. Therefore, it is essential to study about the nucleic acids. Nucleic acid digestion, it is confined to small intestine. In other part of the alimentary canal, there are no nucleases. What are those nucleases? Nucleases are nothing but these are the enzymes that act or digest the nucleic acids. So the enzyme which is acting on DNA or the enzyme which is breaking the RNA, both DNA and RNA are the nucleic acids. The nucleic acid digesting enzyme is called nuclease, or they are called nucleases. And these nucleases are present in the small intestine, so that the digestion of the nucleic acids is confined to the small intestine. The nucleases break down nucleic acids into two components such as nucleotides and nucleosides. What are the points to remember with respect to digestion of the nucleic acids? Nucleic acid digestion is confined or it is occurring in the small intestine. Enzymes which are acting on the nucleic acids are called nucleases. When the nucleases act on the nucleic acids, they produce the two intermediate compounds called nucleotides and nucleosides. These are the first things to remember. Then, what happens when the nucleic acids are exposed to the action of pancreatic juice? So, pancreatic juice, it acts on the food only in the small intestine because the food does not enter into pancreas as well as liver, so that bile juice as well as pancreatic juice, they are acting on the food particles in the small intestine. Pancreatic juice contains two nucleases. In the pancreatic juice, the two nucleic acid digesting enzymes are present. One is DNase expansion deoxyribonuclease. The suffix ase is the indication of enzyme. Then ribonuclease, RNase. These are nothing but the enzymes which break or which act on the ribonucleic acid, that is RNA. Deoxyribonucleases, in short DNase, they break down DNA into deoxyribonucleotides. DNA, in the presence of DNase, it is converted into deoxyribonucleotides. Like that, ribonucleases, in short RNase, they act on the RNA molecule and RNA is converted or broken down into ribonucleotides. When DNA is acted by the deoxyribonucleases, the products are deoxyribonucleotides. When RNA is acted by the RNase, the products are ribonucleotides. So both DNase and RNase are the nucleases which are present in the pancreatic juice, but DNase act on the DNA molecule, they break down the DNA molecule. RNase act on the RNA molecule, and both of them are belonging to the group of enzymes called nucleases, as they are acting on the nucleic acids. Then, what about intestinal juice? Intestinal juice also contains two nucleases. Final action is carried out by the enzymes which are present in the intestinal juice. The intestinal juice is also having two enzymes for the digestion of nucleic acids. They are nucleotidases and nucleosidases. Remember, pancreatic juice contains DNase and RNase, whereas intestinal juice contains nucleotidases and nucleosidases. What is the difference? Nucleotidases act on nucleotides. Just now I used the term deoxyribonucleotides. Ribonucleotides. Nucleotide molecule is acted or broken by nucleotidases. See, the nucleotide, it is acted by nucleotidase. The products of this reaction are nucleosides and inorganic phosphate, because when nucleoside combines with the inorganic phosphate, the resultant molecule is called nucleotide. When the same nucleotide is broken or digested by the nucleotidase, the resultant molecules are nucleosides and inorganic phosphate. Once the nucleoside is obtained, these nucleosides are acted by nucleosidase and the product are nitrogen base and pentose sugar, because the nucleoside is a molecule containing nitrogen base and pentose sugar. To this nitrogen base and pentose sugar, when inorganic phosphate is added, the resultant molecule is called nucleotide. When many nucleotides are added to each other with the help of the phosphodiester bonds, it results in the formation of polynucleotide chain. So polynucleotide chain itself is the nucleic acid. It may be DNA or it may be RNA. So the two more enzymes that are present in the intestinal juice for the breakdown process of the nucleic acids are nucleotidases, which act on the nucleotides, and when the nucleotides are broken by nucleotidase, the resultant product is nucleoside and inorganic phosphate. The nucleoside is broken by nucleosidase and the products are nitrogen base and pentose sugar. Now, the fully digested semi-fluid and alkaline fluid which is present is called chyle. Because alkaline, why the term is alkaline? Because the intestinal medium is alkaline and that alkaline property is maintained by the bile juice. The same reaction here I have given. Nucleotide, it is acted by nucleotidase and broken into nucleoside plus inorganic phosphate, whereas nucleosides are broken by nucleosidase and the resultant products are sugar and bases. This is how the nucleic acids are digested in the small intestine with the help of pancreatic juice and intestinal juice enzymes. The pancreatic juice contains DNAs and RNAs, whereas the intestinal juice contains nucleotidases and nucleosidases. Therefore, the digestion of the nucleic acids is confined only to the small intestine. Then, here it is summary of the different enzymes which are present in the saliva, gastric juice, pancreatic juice and intestinal juice, and on which substrate they are acting and what are their products. If you go through the chart, you will recall the entire thing. You will get the complete information about the enzymes. Salivary glands are present in the mouth. The main enzyme present is salivary amylase. It is present in the saliva secreted by the salivary glands of the mouth. It acts on the starch. The required pH for the action is 6.8 and resultant product is maltose. I have not mentioned about the lysozyme because it is not related to digestion. It is antibacterial enzyme. Then gastric glands which are present in the stomach, they secrete gastric juice and location of the gastric glands is stomach. The enzyme present is pepsin, secreted as pepsinogen inactive form, renin as prorenin inactive form, and another is gastric lipase. The conversion of pepsinogen into pepsin, prorenin into renin is carried out by the HCl. The proteins in the presence of pepsin are broken into proteases and peptides. Prorenin in the presence of HCl, it is converted into renin, and the renin, it acts mainly on the milk protein called casein and it is converted into para-caseinate in the presence of calcium ions. And gastric lipase is lipid digesting enzyme or fat digesting enzyme. Gastric lipase along with the pancreatic lipase, it is converted into triglycerides and diglycerides. Extremely sorry, here it is gastric lipase. So let me erase this one so that the mistake, please you don't remember this as pancreatic. Actually this is gastric lipase. Let me show with the help of arrow mark. The enzyme acting here is the gastric lipase. Extremely sorry for the mistake. Then pancreatic enzymes. The different enzymes which are present in the pancreatic juice. What are the different enzymes present in the pancreatic juice? Pancreatic juice is secreted from pancreas. It acts on the food in the small intestine. The pancreatic amylase, it is acting on the starch and it is converted into disaccharide, that is once again maltose. The protein digesting enzymes trypsin, chymotrypsin and carboxypeptidase are also present in the pancreatic juice. They are acting on the proteins and finally converted into small peptide chain along with the amino acids. So one more thing I need to highlight here. Saliva contains only carbohydrate digesting enzyme. Remember, gastric juice contains 2 protein digesting and 1 lipid digesting enzyme. Pancreatic juice contains 1 carbohydrate digesting enzyme, 3 protein digesting enzymes, 1 lipid digesting enzyme and 2 nucleic acid digesting enzymes. So pancreatic lipase is the principal enzyme in the breakdown of fat molecule where they are converted into triglycerides and diglycerides. Deoxyribonucleases, that is in short DNAs, it acts on the DNA and converted into deoxyribonucleotides. RNA, in the presence of RNAs, they are converted into ribonucleotides. Therefore, pancreatic juice is having all the enzymes, whether for the digestion of the carbohydrate, protein, lipid as well as the nucleic acids. Then moving to intestinal juice. What are the different enzymes present in the intestinal juice? Intestinal juice is secreted from small intestine and this contains enterokinase. Enterokinase is involved in the conversion of inactive trypsinogen into active trypsin. It contains dipeptidases such as maltase, sucrase and lactase. These are the carbohydrate digesting enzymes present in the intestinal juice. Then intestinal lipase. This is acting on the diglycerides and monoglycerides to get the fatty acid and glycerol as a byproduct of fat digestion, and 2 nucleic acid digesting enzymes, nucleotides, tidases and nucleosidases. See, intestinal juice and pancreatic juice, they contain all the types of enzymes. The enterokinase is required for the activation. Carbohydrate digesting enzymes present in the intestinal juice are maltase, sucrase and lactase. Lipid digesting enzyme present in the intestinal juice is intestinal lipase, and the protein digesting enzymes are missing here. Then nucleotidases and nucleosidases. So dipeptidases, I have mentioned here. Yes, dipeptidases, they are dipeptidase, tripeptidase and aminopeptidases. They are acting on the protein molecule. Disaccharidases are nothing but maltase, sucrase and lactase. Dipeptidases are related to the digestion of proteins. So all the types of enzymes are present in the intestinal juice. So this is how these enzymes act on the carbohydrate, protein, fat as well as nucleic acid, and this digestion is completed in the small intestine. So now let us move to absorption of digested food. How it occurs and where it occurs. Absorption is the transfer of end products of digestion from intestine to the circulatory fluids. The colored fluid of the body is blood and colorless fluid is the lymph. After the digestion, the absorption starts. Absorption is nothing but a transfer of end products of digestion. such as glucose, fructose, galactose, amino acids, fatty acids, the pentose sugar, nitrogen base, inorganic phosphate. All these are the end products of digestion. They are transferred from intestine, because in the small intestine only the complete digestion takes place. From the small intestine, all these digested products are transferred or transported to the circulatory fluids to supply to each and every cell of the body, and this is carried out by blood and lymph. In what way this transportation of the end products of the digestion occurs? The absorption occurs by two different methods. One is passive method, without expenditure of energy. ATP is not required to transfer these molecules. Again, in the passive method, there are two mechanisms, osmosis and diffusion. Diffusion is of two different types, simple diffusion and facilitated diffusion. By these methods, the end products of the digestion are transferred to the circulatory fluids such as blood and lymph, so that every nutrient is supplied to body cells through the circulating body fluids such as blood and lymph. So let us concentrate now. What is passive absorption and how it occurs? So passive absorption, as I told, it is carried out without expenditure of energy. That is why the term passive. And in the passive absorption, the two mechanisms are observed. One is diffusion and another is osmosis. And diffusion is of two different types, simple diffusion and facilitated diffusion. In the passive absorption, absorption of nutrients from higher concentrated region to lower concentrated region without expenditure of energy occurs. The molecules which are present in the higher concentrated area, they move towards lower concentrated region without any expenditure of energy. This is called passive absorption. And passive absorption includes osmosis, particularly for the absorption of water, where plasma membrane functions as semi-permeable membrane. Every cell of the human body is lined or covered by plasma membrane, and that plasma membrane functions as a semi-permeable membrane during the absorption of water, and that occurs through the osmosis, and osmosis is a passive transportation of the material. And diffusion is for the absorption of solute molecules. For the absorption of solvent, the type of the process taking place is osmosis. For the solute molecules, the mechanism that occurs is diffusion. And diffusion is of two types, such as simple diffusion. In this, molecules alone can be diffused. For example, small amounts of monosaccharides like glucose, product of the carbohydrate digestion, amino acids, products of the protein digestion, vitamins and electrolytes like calcium. Simply without any particular structure, from the higher concentrated region to the lower concentrated region, these molecules are moving without any specific structure, and such a diffusion is called simple diffusion. Whereas the molecules like fructose and some amino acids are absorbed with the help of carrier ions like sodium. This mechanism is called facilitated transport. Through the protein channel, the molecules are transported. I am telling protein channel, not with the help of expenditure of the energy. Energy is not at all consumed during the passive absorption. Some proteins of the plasma membrane are helping as a channel to transport the molecules, and Carrier ion like sodium is also involved to transport the substances like fructose and some amino acids. This kind of the diffusion is called facilitated transport. Some particular structure is involved for the transport purpose. That is why this is called facilitated transport. Then one more method of absorption, it is active absorption. Then what is the difference? In the active absorption, the energy is used. Absorption of nutrients from the statement is completely opposite. Lower concentrated region to higher concentrated region against the concentration gradient. Therefore, active absorption needs the energy in the form of ATP. See, when the molecules are moving from higher concentrated region to lower concentrated region, there is no need of any energy to push the molecules from higher concentration to lower concentration, because already the molecules are present in the higher concentrated area. They have more energy. They do not need any kind of the energy to move from the higher concentrated region to lower concentrated region. Say for example, water falls. When water moves from tip of the hill to the downwards, there will be no requirement of any motor or any other energy utilizing machine. But when we pump the water from down to up, it requires the energy. Here also the molecules are moving from lower concentrated region to higher concentrated region. So first of all, their concentration is very less. With that situation, they are moving towards higher concentrated region, and this requires additional pull or additional support, and that is used in the form of ATP. What are the examples? Absorption of some amino acids, monosaccharides and glucose. Again, electrolytes like sodium are required for the active transport also. Both of them are taking place depending on the concentration. Sometimes it will be through the diffusion and sometimes it will be through the active absorption. Like this, the molecules are transported to the circulating fluid after the process of digestion. Then moving to next point, absorption of lipids. On this you need to concentrate little bit extra. Monoglycerides and diglycerides and fatty acids cannot be absorbed directly as they are water insoluble. What is the problem for direct absorption? Monoglycerides, diglycerides and fatty acids cannot be directly absorbed as they are water insoluble. So what to do? Bile salts and the phospholipids of the plasma membrane, they convert them into small spherical water soluble droplets called micelles. That is why the emulsification is more important for the digestion of fat. Monoglyceride, diglyceride and fatty acids are water insoluble. They have to be converted as water soluble molecule with the help of bile salt and phospholipid. Now those water soluble droplets of the fat are called micelles. They are reformed into small protein coated fat globules called chylomicrons. Try to remember this term. Once the micelles are formed, they are coated with the protein. That protein coated fat globule is called chylomicron. They are transported into lactales in the villi, because lactale is related to absorption of the lipid. From the lymph, the chylomicrons enter the blood. finally through that lymph of villi, the chylomicrons enter into the blood. And chylomicrons are nothing but these are the protein coated fat globules. See, the same definition, the same explanation is given in the form of schematic representation as well as in the form of steps. These are the water insoluble fat molecules, fatty acids, monoglycerides as well as diglycerides. So what is happening here? Emulsification, so that they are emulsified into smaller water soluble fat globules which are called micelles. And these micelles, they enter into the cell. Now a protein coat is formed around the micelles which are called chylomicron, and these chylomicron, they are absorbed or transported across the lacteal of the microvilli, and finally the chylomicrons enter into the blood capillary. This is how the fat is absorbed in the small intestine. Particularly the lacteals of the villi are involved in the absorption of fat. From the lacteals only, the chylomicron finally it enters from lymph into the blood vessel. So moving to the next one, absorption in different parts of alimentary canal. What are the different materials that are absorbed in the alimentary canal? Because alimentary canal is a long canal or tube structure having mouth and anus, mouth to pharynx, pharynx to esophagus, esophagus to stomach, stomach to small intestine to large intestine, rectum and anus. So all these are the parts. Then question is whether all these parts are involved in the absorption or absorption is restricted to only some parts of the alimentary canal. So let us move to the absorption in different parts of alimentary canal. So where exactly which materials are absorbed? In the mouth, certain drugs are absorbed. In the stomach, water, simple sugars, some drugs and alcohol gets absorbed. Small intestine, all the nutrients including minerals and vitamins are absorbed. Large intestine, water is mainly absorbed. Some minerals and drugs are also absorbed in the large intestine. So mouth, stomach, small intestine, large intestine, these are the parts where all these materials are absorbed. Small intestine is the chief area of absorption. That means main area of the absorption. Why? Because of the presence of villi, the finger-like folded structures, and villi increases the surface area of absorption. It helps or it provides the larger surface area for the absorption purpose. Another reason, its great length, longest part of the alimentary canal, and it is highly coiled, much coiled, except duodenum. The jejunum and ileum are coiled portions. Particularly jejunum is heavily coiled, and coiling region, coiling part or coiled structure, slowly it transfers the food to next part. During that only the complete digestion and absorption of the materials is carried out within the small intestine. Absorbed materials are incorporated into the tissues for their activities. This is called assimilation. Try to remember this definition. Once the materials are absorbed into the tissues, how they are transported? All the digested food, the end products of the digestion, are transported to body cells through the blood and lymph. and this is carried out either by the active process or by the passive process. It may be the osmosis for water absorption, it may be diffusion for the absorption of solutes. In one or other way, the materials are absorbed. Absorbed materials are used by the tissues for their activities. They are gathered, they are incorporated, they are transferred into the tissues for their activities. This is called assimilation. Undigested substances like plant fibers, some quantity is digested and some quantity left or remains undigested. Dead bacteria which are killed by HCl or by lysozyme, they form the feces. Feces means undigested food material. This feces or undigested food material enters the cecum, which is the part of large intestine, through the ileocacal valve, because ileum of the small intestine is connected to cecum. In between cecum and ileum there is a valve called ileocacal valve, and through this valve only the feces or undigested food, it enters the cecum, and which prevents the back flow of feces. Means the ileocacal valve, it prevents the re-entry of undigested food into the small intestine. Once the undigested food called feces, it is transferred into the cecum, its reverse movement or back flow is not possible, and that is prevented by the ileocacal valve. Then feces are temporarily stored in the rectum and are eliminated through the anus. This is called egestion or defecation. Defecation is the term used for ingestion and egestion. The removal of undigested food from the body through the anus is called defecation. This is called defecation. Temporarily that undigested food, which is called feces, it is stored in the rectum, and finally that feces is removed through the anus through the process called defecation. These are the terms: assimilation, where the absorbed materials are incorporated into the tissues for their activities, it is called assimilation. Undigested food from the small intestine, it is transferred to the cecum through the ileocacal valve. The ileocacal valve prevents the back flow of feces. The feces is temporarily stored in the rectum and finally it is removed through the anus. The process is called defecation. Then the last point of today's video, after assimilation, see what happens finally to the absorbed materials after assimilation, where the assimilation is taking place in the tissues. The various nutrients are divided into 4 categories according to their requirements, because the food contains all the variety of nutrients. After assimilation, these nutrients are divided into 4 categories. What are those? First category, energy producers, and they are nothing but carbohydrates and fats, because their combustion or breakdown produces energy for different activities. Bodybuilders are nothing but proteins, to produce different body structures, body parts, because maximum in our body is made up of proteins. Metabolic regulators, vitamins and water, minerals. Then hereditary substances, see, they are nothing but nucleic acids. So after assimilation, depending on their requirement, the nutrients are divided into 4 categories: energy producers, bodybuilders, metabolic regulators and hereditary substance. The bodybuilders are the proteins, the energy producers are carbohydrate and fats, Metabolic regulators are vitamins and water minerals, and hereditary substances are the nucleic acids. Then the energy value is specified in kg, kilojoules per 100 gram or 100 ml, and here is the caloric value of different energy molecules. The caloric value of the carbohydrates is 4.1 kilocalorie. The caloric value for the carbohydrates is 4.1. For that of fats, highest it is 9.45 kilocalorie, 9.45 kilocalorie. This is the caloric value of fat, more energetic than that of carbohydrates and proteins. Then proteins, the caloric value is 5.65 kilocalorie. I have used some specific color for the organic molecule and the related energy value so that it will be easy to remember. For the carbohydrates, the caloric value, carbohydrate caloric value is 4.1 kilocalories. For the fat, it is 9.45 kilocalories, and for the proteins, it is 5.65 kilocalories. The highest caloric value is obtained with the digestion of fat molecules. So this is about the caloric value of different energy compounds that we consume through the regular diet. So these are the things that I wanted to explain in the today's video. So hope this explanation is clear for you. In the last part of this particular chapter, I need to explain about the digestive system disorders, and I will continue with the multiple choice questions on the digestion and absorption in the next video. And you need to do is just to go through all the parts of digestion and absorption so that you can solve the questions more easily. Hope the explanation is clear for you. Thank you.